So my name is Dr. Kate Connor. I'm an assistant professor in physiology here in Trinity College in Dublin, and I'm a cancer neuroscientist with a focus in neuro-oncology. So I really got into science because I had some really amazing teachers in my secondary school who just really inspired me. And I went on to do a degree in medicinal chemistry in Trinity College Dublin, and that really is where my love of science grew. But I took a bit of a side turn and I wound up doing a PhD in breast cancer pharmacology in University College Dublin. And once I finished my PhD, I got the opportunity to start with a postdoc in the Royal College of Surgeons, which was focused on neuro-oncology and better models, developing better preclinical models for studying neuro-oncology and interventional therapeutics. So I spent about 7 years in the Royal College of Surgeons studying brain tumors, specifically glioblastoma, and working on projects which aimed to develop improved therapeutics for these patients. This really was where my love of neuroscience came from. We worked in the brain, but we didn't really think about the neurons. And so when the opportunity to work with Professor Mark Cunningham in Trinity came up to work on a project which kind of bridged both cancer research and neuroscience, I jumped at the chance, which is when I became more of a cancer neuroscientist and I developed my skills in electrophysiology. So I have a couple of different research strands. One of them is focused on understanding what role mast cells play in the driving of brain tumor-related epilepsy. So this is quite interesting because these are cells that are normally, associated with allergic responses, but because they can be very pro-inflammatory, I'm currently trying to understand, can these cells be modulated to try and improve patient outcomes when they have brain tumor-related epilepsy? And I'm also working on developing a number of clinically relevant and more faithful preclinical models of brain tumors. So I work with developing, these are all orthotopic, so they're cells implanted in the brain. They have a fully competent immune system, which allows you to study various therapeutic interventions. And also we're developing translational imaging for these models so that we can fully understand them and try compare to the patient scenario. So a lot of the work on developing better preclinical models is aiming to have better clinical trial outcomes in the long run. And ultimately, a lot of the work then is focused on identifying novel therapeutic vulnerabilities within the glioma, specifically glioblastoma microenvironment, which may be targeted to help treat both the tumor progression, but also the comorbidities with these diseases, such as epilepsy. So one of the key things when developing a good preclinical model is ensuring the clinical relevance of this model. So we have developed protocols for in vivo EEG in brain tumor-bearing mice, and we're able to longitudinally record their seizure activity and their electrical activity over time. But we also want to be able to take the tissue from these animals and study it in vitro environment in the ex vivo setting. So we can, use various therapeutic interventions to see the response of the tissue to this. So through brain slices, we're able to take tumor samples from our animals, which have a peritumoral zone. We can create brain slices and we can do local field recordings or patch clamp recordings on this tissue. And this allows an ex vivo electrophysiological characterization of a model, which we have the in vivo characterization, but it also then allows further downstream, for example, immunocytochemistry, proteomics, transcriptomics. So really with the ex vivo brain slices, we're able to take a step towards understanding the mechanism of what's happening in these preclinical models to help us understand better resistance mechanisms or response mechanisms that we see in vivo and then ex vivo. So we use 400 micrometer slices, which we cut at 0.1 millimeters per second. And we've optimized this for being the best for our local field recordings and allowing further slicing downstream for our stainings that we do. One of the things that we have found helps really, really well, especially in brain slices where you might have a tumor or you might have some sort of damage to the brain tissue, which makes it more fragile, is that we use agarose blocks to help support our tissue when we're cutting. So what we do is, depending on the brain region you want, we orientate our brain and we glue it to the chuck. But then we take small blocks of agarose between 2 and 3% concentration, and we glue them next to the tissue. And what this does is it gives you a support to help with the cutting of the tissue, and to stop it from fragmenting or getting caught as you're cutting. Because when you have a tumor, often they can be vascular, and this can be very difficult to cut cleanly. So if you're ever having difficulty with a difficult brain region or a fragile tissue, these agarose supports can be really, really helpful. Also very helpful with human tissue where it's often quite fragmented on the edges. Some of the interesting findings we've seen is we have taken brain slices from normal, healthy wild type animals, and we've treated them with 4-AP to induce seizures. And we've begun to treat with various modulators that mast cells produce. And we've been treating with various combinations of these modulators to kind of mimic what mast cells might be doing when they enter the peritumoral region. And we've been able to see some really interesting patterns of response when we treat these seizing slices with serotonin and histamine, and we're able to see quite an interesting inhibition of the seizure activity at quite low doses of these modulators. So on the flip side, we're now beginning to treat with some more pro-inflammatory modulators that these mast cells can produce, such as IL-6. And we're going to be assessing what the impact of that will be on in this model. So ultimately, what we're able to do with the brain slices is really pick apart the mechanisms of what the various mast cell modulators might be doing. And the next step will be to do that in the brain tumor bearing slices. So in the next 5 to 10 years and moving forwards, I think what's going to happen in the field is there's just going to be a huge increase in interdisciplinary work. As a cancer researcher, we often forget there's other cells doing things and driving the disease. And I think now that we really understand how important neuronal crosstalk is in driving malignancy and driving cancer progression, I think there's going to be a huge explosion of research into this field, which is really going to help us treat patients better because we're going to understand better some of the driving forces underpinning the tumor and also tumor associated comorbidities like epilepsy. And also through this understanding, we'll begin to maybe understand the impact that cancer treatments might be having on normal neuronal functioning, which will ultimately allow us to treat patients better and ultimately improve patient outcome and patient quality of life.